y'all. So my friend Joshua Birkebak, who has a PhD in fungal systematics, taxonomy, and evolution, was kind enough to sit down and walk us through his process of creating phylogenetic trees. Hopefully you enjoy this impromptu little workshop as much as I did. Okay.
All
Okay.
right. Yeah. So I put links to two programs. I mean, I assume you already have Eugene and anyone else can probably get that. But then there's also Raxaml GUI, which is the program that I find to be the
Yeah.
easiest and sort of pretty and pretty honestly thorough, much more thorough than sort of Eugene's sort of built-in
Mm.
maximum likelihood tree methods. And yeah, it's a good program. We'll go over it for multiple reasons. And then Fig Tree, there's multiple different tree viewing software. It's the one I use. It's not perfect. I'm happy to hear some alternatives, but it's a pretty quick. I don't know. Again, it's just sort of what I'm used to. We'll see if people like it or not.
You're fine.
Almost
Where's
not.
I don't see the download on the Fig Tree page. I got the other one downloaded.
On the
Is it
right,
is it
sort of
just
in that little
this?
window, it says binaries are
Oh, it's
available
just the
from
GitHub repository.
GitHub. Yeah.
Uh,
Which, again, as you will learn, is sort of the limits of I'm not a particularly computery person, and some of the stuff I may do, somebody who really understands computers might be like, oh, there's a way easier way to do this, or oh, we could automate this workflow, which, David, I'd love to see things like that. And yeah, and
yeah.
like if we have time too, especially, it's one thing to have a FASTA file and make a tree. It's a whole nother thing to get data out of GenBank and Micromap, where I have ways to do it. I'm not going to claim that they're necessarily optimized, but they're ways that I can do. So we'll probably go over that maybe after we just go over that you already
Yeah.
have your data. And then we can
So,
maybe talk more about how to get data compiled.
right. Do I download Figtree version 1.4.4 zip.zip, I assume?
1.4.4 is what I have, yeah.
Yeah, okay, cool. Let me get these set
All
up.
right, I guess I should probably share my screen, right?
Mhm. Yeah, I'm just installing these real quick.
So, spurred on by your
It's a rabbit hole, and like, I'm probably not the person that's actually qualified to go down this rabbit hole further than editing. But
let's allow. All right, is it showing?
yeah, here, I'll just install these later.
Okay. Basically, I'm going to go ahead and delete some of this test stuff I already did just to make sure. But excuse me, I pulled all the
Mm,
uh, Pulver Lepiota data. So I got everything that was on GenBank and everything that was in Micromap,
mhm.
which actually I would love to talk to some of you about whether I'm even doing this the right way or like the easiest way or not. But here we've got our FASTA file, right? Like everyone, hopefully recognize it's just literally a definition line followed by the string. And this is pruned down to, this is all just ITS. I've got another example if we want to look at multi-gene, which we probably do because that's one of the things that's amazing about Raxaml is it basically automatically makes a super matrix for you where you can like really easily do a concatenated data set. But we'll start with just ITS. So here's everything from there. And I mean, this will be a pretty quick walkthrough, especially because it's a small data set, which is like a great example. Oh, of course, it's opening up on this one. All right. You can see it, right?
Yep.
Okay. So we'll join our sequences together into an alignment. We'll update Eugene later, because it always likes to want to update. Go ahead and align using MAFT, which is sort of the fastest and pretty accurate. We've got a relatively clean alignment. Um, there's some weirdness in there, but there's some weirdness in most fungi. Always go to the 5.8s to give it a look through to see if there's anything misaligned or really weird. You'll always sort of be able to tell by glancing at the 5.8s, right? Because it's pretty highly conserved. Like I said, we are kind of jumping in here. We already put this data set together and cleaned it up a little bit. Maybe we'll want to kind of go and like make a new one at some point. So what I'm going to do here is I'm probably going to trim it to, usually as a rule of thumb, the point at which it's like 50% gaps, I'll just remove that. The ends of Sanger reads, just like the ends of nanopore reads, are messy. They're kind of noisy. You can still see here too. That could be a real difference. That could just be noise. This is honestly probably noise. If we were really concerned about it, we could probably trim it back to here. Maybe that G. Yeah, maybe that C. See what I'm talking? There's just like weird gappiness that's probably not real. It's just probably from poor resolution. So I'm going to go ahead and trim it just a little further. Again, it's sort of like, here's an obvious mismatch here as well, right? You can see that that A probably needs to move over, and then it would be GGA, and it would line up with everything. If your data set is like an ITS data set, it should have lots of other differences where occasionally having just a little bit of noise in there hopefully won't overcrowd all of the real signal you have through the rest of your alignment. So you don't have to fret every single little thing. Another thing that I'm going to do here too is I'm not going to fuss over the alignment too much. Sometimes, in the same way, you'll see weird things. You could go, oh, what if I scooted this over? I could maybe eliminate some of these gaps. From a reproducibility standpoint, you might not want to do that. You just literally, that way you could just say, hey, I did what the computer spat out and I ran it. And unless you're looking at it going like, this is so ugly, I don't think there's real data here, just ignore little errors here in there. And then here we're going to trim. See, one of these is, an ITS and LSU sequence. So we've got a big, long stretch where we just have LSU. So we're going to remove that. So now we've trimmed it down to pretty much just the ITS and the 5.8S. At this point, honestly, we've got an alignment. One thing I'll do sometimes, and I'll just show it here, I wouldn't do it for a data set this small, but I may make a real quick neighbor joining tree, like just really quickly to run it and make sure there's nothing sitting out on like a stupid, crazy long branch. When you have a really big data set with a couple thousand potentially sequences, you might do this real quick. Neighbor joining is like one of the oldest, most basic and simple methods of tree analysis. And it's really quick. It doesn't really give you good topographies, but it's really quick and it'll help you to sort of identify anything that's crazy. You might have missed something that needed to be reverse complemented. You might find something that's like a fusarium or an aspergillus, like, a contaminant that got sequenced. Nobody did quality control on their data, and you should be able to identify those here. But everything looks fine. We can just go ahead and remove it. So now what I'll do is I'll export this alignment as I'll uncheck the add exported document to the project because that just muddies things up. And then I'll make sure I'm in this folder. I'm just going to name this ITS_align, or align if I can spell. FASTA. Wow. This is one of those things where I promise I can type except when people are watching,
Yes.
So now we have a FASTA file that is literally just the alignment that we saw. If you open it up and look at it in a text editor, for example, it's the exact same thing where you have just the definition line and your string of nucleotides, except now it has the gaps so that they'll all line up with each other. Nice and simple. Let's go ahead and open up RaxML GUI. Sorry, everything is opening up in the wrong window. So this is what the program should look like. Pretty simple. We can drag our alignment up here. So now we've got a couple different things going on. So first thing we're going to do is run model test. So model test is going to compare all the different nucleotide substitution models that are available, and figure out what is the best fit for our data set. So it's looking at transitions and transversions, proportion of invariant sites, and sort of the rate heterogeneity model. Not going to get too much into detail of this, but basically the most simple model is Jukes-Cantor, which is basically a change from an A to a T is just as easy as a change from an A to a G, or an A to a C, and a C to a T, and a C to a G. So basically, they're all as likely to change nucleotides to each other, right? We know that that's not true. Just from looking at sequences, when you normally see degenerate nucleotide symbols, they're usually like Y's and R's, because you're usually going to have a switch between your purines or your pyrimidines because they don't disrupt the structure of the DNA too bad, and they're a lot easier for sort of the molecular machinery as it's copying a genome to make. So we kind of know that that's not the correct model for real life, but it is sort of the simplest model. And then essentially, your most complex is GTR, which is that every single transition and transversion between every nucleotide has a different rate, which may or may not be true, or maybe is technically true, but there's sort of a question in this, right, where it's like, as you add complexity, you can risk over parameterizing your models, basically. So sometimes it's not worth it, even if it might possibly be true or like more correct. But basically, this is going to search through all of these possible models and pick the one that's the most likely for our specific data set. Not for these species, not for the genus, just literally for this specific data set. So it can change, depending on what you add. It's probably going to run really fast. So yeah, so it's going through and it's calculating a likelihood score for all of these different models. It's a really small data set, no time. It decided that we want TMV plus gamma and not proportionate of unvariate size. So basically, it'll auto-populate up here with what it has decided is the best fit model. So you don't have to do that or anything. You just kind of let it do its own thing. We're not going to mess with any other partitions or alignments right now. And then what we'll do is, what I usually do is I'll go ahead and just do a maximum likelihood tree inference with a single run. This is a test. This is just a test run. Or if you're just doing a quick and dirty tree, especially if you have a really big one, you'll just go ahead and just do one. No need to overcomplicate it right now, because what you may find is you run it and you get some craziness because you missed a couple bad sequences. There's a couple things that need to be reverse complemented. You could have waited 3 hours and then gotten something that you realize, oh crap, I got to go back. I got to refine this further. There's a bunch of misidentified stuff. The other thing we're going to do with this data set is I included an outgroup of two cystolepiodas. Well, two melanophyllums technically, because cystolepiota should be melanophyllum. So we'll select these as our outgroups, and that's what's going to root our tree so that we're sort of comparing pulveralepiota in a way that we think, or we're trying to get the closest to sort of, or closer to what sort of a real tree is. Obviously, we have a single locus ITS, don't read too much into that, but it sort of gives the tree a direction from a root moving upward. Just to be pedantic, I usually like to set the random seed to 1 because the number 1 is just as random as any other number. So I usually just set it to 1 because why not? And otherwise, we'll just kind of let it go. And again, this is probably going to run in no time. There we go. We have our maximum likelihood tree. So it was really quick. We're going to look at our best tree. We only had one run, so it's just going to be the tree we got. Double click, and that will open up here in Fig Tree, which is just the tree visualization software. And then if you push Control U or go to tree increase node order, I just like that shape. That's the way I was sort of taught, from bottom to top. You can also view your tree in decreasing node order, which you'll sometimes see in some papers. I think this is literally, doesn't matter. It's the same information, just displayed top to bottom, bottom to top. I'm a bottom to top kind of person. That's just the way I was raised. So we can look at our tree here real quick, and we can just sort of do a sanity check. We'll see we've got our outgroup here, which is good. It should always sort of be at the root, especially since we literally told the software it needs to be the outgroup. Then we sort of have two main clades. Your clade with the most species is always going to be on the top. That's just the way the software decides to order it from few, or not species, but tips or sequences. So always going to start with the least and move its way up to the most. And here we sort of have two clear clades. We've got Patassiformis povirulenta, question mark, and then we've got a whole nother section of interesting things here. We've got something from China, something from Australia, something from India, something from China and Australia, potentially named Oliviere, and another species from Hawaii. At this point, I can't help but start thinking about stuff because I like thinking about things like, huh, that's interesting. Do we have one species in the North Temperate and almost all of the rest of these are like paleotropical? That's kind of interesting. Maybe we had one sort of long distance dispersal event or something like that, but who knows? We don't necessarily have a ton of sampling. Here, we'll just kind of give it a quick look to see. We've got some interesting structure here that I don't quite know how to interpret yet, but we have a cluster here of some of the haplotypes, maybe CA03 possibly. We have the two European sequences of Elsas that are of Pulverulenta from Europe. And then we sort of have another mess of stuff up here, which is some of the other haplotypes. So I
If you
took the haplotypes
pull,
and put them in separately.
oh, you took the haplotypes and put them in separately.
Yeah, so
Gotcha.
I have both the consensus and each haplotype.
Heard.
What I
Okay.
want to do after this is I want to cut out all of the consensuses and just look at the haplotypes because I think that'll tell us something slightly different. Because what we have here is we have some of, for example, here, HVS, we have some of Helen Smith's collections that Stephen sequenced that are highly polymorphic because they're probably a blend of the two haplotypes, which means that as far as the tree structure goes, if we're going to have the two haplotypes and sort of the intermediate, we're going to get a little bit of a mess here because some of the sequences are going to look like both and it's going to have trouble placing them. Before we get to that, we'll go ahead and run a tree with bootstraps just to sort of see what we got. I'm going to run transfer bootstrap expectations plus consensus. I'm going to set it to 20 runs. I would normally do 20 runs for anything. And then what I would normally do is I would do auto MRE, which is where Raxamel itself will decide how many bootstrap replicates you should do based on how different the results it has. So sort of once it gets a good distribution of bootstraps, it'll tell you, hey, you've got enough, or it'll reach 1,000 and it'll stop there. Let's just run 100 for this since this is still just kind of informal. If I were running something for a publication, I would choose the auto MRE, and sometimes these run for 8 to 12 hours when it's like a really big data set. This is going to run in no time. So we're running our 20 trees. So it is calculating 20 different trees from 20 different random starts, and it's going to pick the best one. And then here it's running through and it's doing our 100 bootstrap replicates. So the bootstrap replicates are essentially, it's already done. The bootstrap replicates are taking 70% of your data and running the tree again and just looking at the topology, not the branch lengths of how different they are from each other, but whether they cluster with each other compared to the other things or not. And it's a not technically statistical way to get a rough idea of sort of support, but it really all it does is it tells you how strong your data is. It doesn't actually tell you like the probability of the topology, right? It just tells you how much signal is in your data and whether you have a lot of signal or a little bit of signal. Um, which is still informative, but don't think of it as a statistical test of like topology. So here we're now going to open up this file, which is called .support.tree. So that has the best fit tree, so whatever the best tree was, and it's put the support values from the bootstraps on there. And it's going to pop up with this dialog where it's going to want input. We'll just hit label. It doesn't really matter. Sorry, I keep having to drag it over here. Here we have our same tree. Gonna sort it to be ascending, and then we'll click branch labels. And it's going to default to branch times, which is just telling you the branch length. Not that interesting because you can literally see it, you know. But if you wanted a number, you'd have a number. And then you can change it to label, and now we have our bootstrap values plotted on the tree, sort of subtending the node. So what it's telling us here is that one or 100% of the bootstrap replicates gave this clade together, which it has to, so that's a little bit of a fact because we told it, I need you to have these two things be the outgroup. So this better have 100%. If not, something's going wrong. And then what we see here is that it's very confident that it's not very confident. Our data is very strongly showing that this clade they're all most closely related to each other and separate from this clade. So we have 99.95 and 97.5. So very good separation, very clean, very strong. If we zoom in here, start to see that things are definitely a bit more messy if we zoom in on this group, which we expect because we have consensus sequences that sort of bridge the two haplotypes. And so we don't honestly have particularly strong bootstrap values here. They're in the 70s, which is like, you know, it's not garbage, but it's not really strong support. So basically, based on just this, we see that there are a lot of differences. Doesn't necessarily help us necessarily sort this out. We can see there's sort of maybe a cluster up here and sort of a cluster here, and we've actually got these two European sequences well supported together. They're almost identical, that those two are sort of unique. They're not integrating with these other sequences. But the placement between these two different haplotypes is not super strong. Um, any questions about this? Sorry, I realize I might have speed ran through that a little bit.
No, I follow.
Yeah.
I have a quick question, if this is the right time to ask, is I'm not exactly sure how to choose the roots, and I would be curious of how you choose what to root things.
Oh, how I picked what to root it with?
Yeah.
Yeah. So normally, like to be honest, depending on what I'm doing, I may not use an outgroup to root at all. It sort of depends. So what rooting something with an outgroup does, like I said, is kind of give you that directionality of sorting your clades out to where you're trying to reconstruct the evolutionary history compared to compared to a related group. If you're just interested in clustering species together to see if these, three temp codes are different or not, you can just not use an outgroup at all in what we call midpoint rooting. And then you're just looking at the relative relationship to each other. But rule of thumb for an outgroup would be you want two or three representatives of the most closely related group that's outside of what you're interested in. So if it's the genus Cymosobe, you might pick Tuberia. If it's the Cymosobe centunculus group specifically, you might pick Cymosobe seriolata and another species of Cymosobe to be your outgroup.
Yeah,
You
so
don't
like if
want
we're
them.
so if we're like looking at Amanita, like a different Amanita section.
Yes, yeah. If you're looking at a
Yeah.
section of Amanita, you might look to find what is the closest related section of Amanita.
Hmm.
You could use an even more distantly related group, but the problem is the further away it is, the more gappy and noisy your data gets, and you might lose signal in all of the noise of having some of this. There's definitely, I've seen a lot of papers recently in certain, especially in certain groups where they use like 30 different outgroups and they're across multiple things. And my honestly, my opinion is that at a certain point, you're actually just diluting the signal in your data. If you are looking to place something within the order Agaricales, though, yeah, you want to include a whole bunch of stuff to see where it's going to show up. But if you're just outgrouping, yeah, you might want to have a couple different outgroups. You might want to add like a Lepiota sensu stricto or something, you could.
Okay.
It's it's not super important for the question we have, which is what's going on with this Ptaciformis group. If we were
Yeah.
wanting to really understand the evolution of, a couple different genera in, Verrucas floraceae or something, then we would want to do that. And we'd want to have multi-gene data and stuff like that. But um, yeah, it's really not important. We could run it without it. And what we would do in this case, especially if we just ran it without the outgroup, especially since it's so simple, is it would be as if we just only had this tree. We
Yeah.
would midpoint root it and it would split it into the top half and that little bottom half there. So
Okay. Thank
and
you. That was very helpful.
yeah. Right, let's think about maybe we can actually at this point, before we sort of go into really like manipulating and changing the data around, maybe I'll show you how to do a multi-gene tree real quick because that's super handy. And this is that's not a real name. This is just sort of a placeholder, but I have here for Pluteus picked out ITS and TEF1, and we're just going to go ahead and do a quick multi-gene tree for that. And actually, let's go ahead and look at let's remake these alignments because there's some interesting stuff to be made here. You know what? Redo or zoom, because I do want to keep those original ones just in case something funky happens. So we're going to copy, paste two items. Okay, so ITS is going to be the same. I mean, I guess I don't really need to show you the FASTA, but it'll be the exact same thing here, where we've got in this case, one specific sort of target species, and then sort of a bunch of representatives of other groups of Pluteus, other clades that we're sort of looking to place an unknown Pluteus into, which is the one that says this one. So clearly indicated. And then with our ITS data set, I'll also open up the TEF1. So TEF1 is a protein coding region, which is going to be a little bit different. I mean, the structure of the data is going to be the same, where we've got, you know, exact same situation where we've got sort of our definition line and our sequence. One thing that's very important when you're doing a concatenated data set is that the definition line has to be identical between the different ones, or else the software won't know that you're saying this is the same specimen. I need you to match this with this. This is a problem where a lot of people, or it's like kind of easy to use GenBank numbers, right, as your sort of number here, but that works for single gene regions. You know, if you're trying to concatenate, your ITS and your TEF1 and your LSU and stuff all have different GenBank numbers, so it's not going to be able to match those up. So what you got to do is then you got to sort of go figure out what is the specimen voucher number, or, you know, or just pick an arbitrary, whatever. You just need to make sure those are identical. And we can look at how I do that differently and some of my pet peeves around how people put that data into GenBank, because people for the same specimen will use different punctuation, use a space sometimes, not use a space sometimes, put extra information. So you can't literally just put it all in one data set. You have to then manually fix the fact that sometimes it's under specimen voucher, sometimes it's under isolate, sometimes it's under biomaterial, sometimes INAT is spelled out to iNaturalist, sometimes it's www.inaturalist.com/observation/inat number, you know, right? Like it's always formatted differently because no one ever does anything the same. And before I get too high on my horse, I've looked back at my own GenBank submissions and I have done the same thing. So
I almost did that yesterday for the Penniless. I actually tried instead of the temp code FLO2, I tried putting Oligotrophus, but it didn't let me. So it went in with continuity.
just be consistent and have as much continuity as possible because it helps life. So all right, so since we have these two, and I've already gone through and sort of painstakingly curated these and made sure that the names are identical, and then invariably there's one that is one space off, and I realize it after I've made the tree, and I've got to go back and fix it. Hopefully I've caught it on this one because I've played with it a couple times. We'll just drag both of them into here. Oh, no, we don't. We have to align them first. That's an error that you get if you haven't aligned your sequences, so pretend like that didn't happen. Go ahead and open this up and mute Eugene. Join into a multiple alignment viewer. Ah, here it is. Here's our ITS data set. Align with MAFT. Do that real quick. And we'll look at our ITS. Pretty reasonable. I mean, it's kind of messy. But we're looking at a whole genus. When we're looking at Pulverlepia, I mean, we're looking at a handful of species. Honestly, this alignment's it's ITS, it's a little noisy, and honestly, some of this is just junk and noise. We'll see what the tree says, but we're not going to fuss around with it too much right now. But this is not very well aligned. This is an example of something that's kind of struggling to make sense of how quickly ITS evolves here in this region. So take that for what it is. But hopefully here, we've got a lot of... Oh my gosh, you know what? Never mind. Okay. Sorry, scratch that. We're going to start over. I remember that because that's way too noisy and I was very confused. Last time I did this, that is TEF1 and we were looking at the introns there. And this is ITS. I accidentally changed the names on these. Let's double check this. We're going to open up what should look like the real ITS now, and we'll make sure it looks like it should. God, I just really need to do that. Okay. I remember being really, now all of a sudden it came back to me. I was looking at it going, what happened? I realized I misnamed them. This looks better. Here's our 5.8S. We've got our big, mostly the same, very few changes region. Here's ITS1. It's noisy, but it feels like there's a lot of decent signal in this data set, even though there's a lot of gaps and stuff. And ITS2, sort of the same picture here. There seems to be a lot of real signal. It's not just making total jumbled noise out of it. That section maybe a little bit. But before we get in the, without getting in the weeds too much, I'm gonna go ahead and trim this here. Trim the noise, and also just don't add a bunch of nucleotides to the tree, for the software to have to handle when there's not a lot of information to be gained from all that extra stuff that only has a couple different representatives in it. Now we're going to go here and we're going to change to this folder. Please don't pay attention to my folder management skills or lack thereof.
You should see my computer. It's chaos.
Okay. Well, maybe I shouldn't be so hard on myself, but I feel like I always feel like it's a mess. Okay. Here's TEF1, so we'll go ahead and align this again, and this makes more sense. Yeah, so what we have here is we have protein coding, protein coding, protein coding, and then we hit an intron. An intron's just a bunch of noise. Protein coding, very clean, no gaps or very few gaps. If you do have gaps, which I don't even know that we do, in the protein coding region, they should be in triplicate, or they should be in triplet, right? Because sometimes they lose whole codons, but if you have a single nucleotide indel frame shift, you're going to completely mess up your protein, right? So you will see gaps sometimes, but they'll almost always be in sets of 3. It's a good way to kind of spot check and make sure things don't get crazy, except for your intron regions, which is totally, you kind of expect to have these junky junk regions. Another thing that's kind of nice with protein coding regions is you get to see this sort of the genetic code at work here, where you could very easily and automatically see your first, second, and third codon positions. So you got your first, the most highly conserved, your second, which is only slightly less conserved, and then you've got your third wobble position, right? And you can literally see this pattern of similar, similar, more different, similar, similar, more different, similar, similar, more different. And that's always just it's kind of nice to see that kind of stuff, Science is real. What we're going to want to do with this is we're definitely going to want to trim out these introns, because we're going to tell the software this is a protein coding region and I have these codon positions, and those are evolving completely differently than introns are evolving. Introns are not totally junk DNA in the way that we sort of might have used that outdated term. There's some structure to them, and there are some requirements of what makes an intron, but in a lot of ways, and in some ways, there's a lot of free evolution that's not constrained by anything. So what we're going to want to find is our GT for the start of our intron position here, and it's going to end with an AG, and there should be, sometimes there'll be multiple GTs that could sort of be near here, but this one, there should be no nucleotide variance unless there's poor sequence quality. If you have like 99.9% GTs and maybe there's one GC, it's probably sequencing error, but it should be basically all GT and then end in something that's all AG. And then we get back to our sort of codon, our triplet sort of pattern here, with very limited evolution on each flanking side of the intron often. Not always. Sort of depends. You can't really immediately see the triplicate pattern because these first 3 nucleotides are all identical. But we'll literally just sit here and remove it. We'll trim it out. Same thing here. We're going to have, here's our GT that we're going to start with. We're going to scroll over here to our AG, and it's pretty tidy. And remove it. And then here we sort of have a decision to make. We have some of these sequences that are much longer reads of that's just random noise. Ignore. Sometimes the alignment software has really weird problems with it'll put one weird little flanking chunk way away when it really should be pulled all the way back that way. We're going to exclude this all anyways, especially because this is all one species. So we're not getting any really useful information from having all of this long string of information. And these overlap a little bit, but when you have a ton of extra data for only a few sequences, the tree software, the tree algorithms have a trouble with that. So you don't want to have stuff be too imbalanced, even if you do have data for a bunch of things. It's not good data, or it's going to cause some algorithmic noise. So I'm going to trim it pretty conservatively all the way back here. And then here we can see our nice little triplicate pattern, right? And honestly, so here's just a subjective call I'm going to make as well. See how this one Pluteus flamans is honestly, it starts really, really late. And gosh, I don't even know. That's like 150 base pairs. That's just going to really probably limit the signal that we're going to have in our tree and just contribute noise. It's going to like artificially decrease our support. So I'm going to go ahead and remove this one. Who needs it? It's not contributing very much. With ITS, there's a similar sort of judgment call to be made with sequences that are only ITS1 and ITS or just ITS2. Sort of up to you. It depends on what I want out of the data set and how much data I have. And, if I have a ton of things of what I'm pretty sure that species is, I'll exclude them. If it's an ITS1 of a type that's, pivotal or, useful, I'll leave it in. But what you do want to be really careful about is data sets where you have a bunch of things that only have ITS1 and a bunch of things that only have ITS2, because it can't compare between them, right? Those two sequences cannot be compared to each other. Now, they'll each be compared to a full ITS sequence that might be of the same species that'll bridge across them. So it might be fine, might not be. It really depends on the exact data set you're working with and what you're trying to get out of it to make that decision of whether you want to leave in a bunch of half sequences. Always make sure you get rid of really short sequences that are only like 250 or base pairs. You don't want anything that short. The alignment software sometimes can't even figure out how to play short sequences like that. And also, if it's Illumina generated or something, it's just going to be riddled with noise. I mean, it'll look like you're looking at a nanopore alignment almost. I mean, not as gappy as a nanopore alignment, but it's noisy. Individual Illumina reads are chaotic. And you'll see them, you'll make something, you won't realize it's an Illumina read. You'll look at the alignment and go, why is this one just like so different from everything else in like random ways where everything else is identical, but it has a G. Everything else here is a G, but it has a C and a T. You'll know when you see Illumina reads usually. All right, so we have our FASTA alignment. We'll go ahead and export this. Well, I'll show you what happens if you don't uncheck that. You really don't have to, but it just kind of makes Eugene have a fit. F1.
I'm realizing the Eugene tree building software isn't necessarily the strongest.
For some reason, if you add to project, it just like puts every single sequence in here.
Jeez.
There's nothing like wrong with it, but it's just doing something that I don't know why it's doing that. And I just check the box. Especially if I'm like working with a bunch of alignments, it just gums everything up. So that's why I usually check that box. We can go ahead and close these. Yeah, Eugene's tree building is like, elementary. It's not useless, but it's sort of like you can do a quick tree with it. The other thing is that it takes significantly longer than Raxamel. It's not optimized to run, in parallel. And so it's kind of noisy. So here we're going to do the same thing. We've added our ITS sequence. We're going to run model test, and we're going to let it decide based on this data set what our best model is. And then we're going to go here and click add alignment and find wherever we were here. We're going to go ahead and add our TEF1 alignment. And then we're going to tell it that we want to edit the partition. So since we have a protein coding region here, we want to tell it that there is a first codon position, a second codon position, and a third codon position. Because what it actually needs to do is it needs to treat them all separately, because the third codon positions are going to wobble a lot, right? And that's great because it gives us signal, but a change in a third codon position is not the same as a change in a first codon position. We don't have very many of those, and those are, on average, typically more indicative of a more serious evolutionary change that's much less likely to happen because it's probably causing a change in the protein, which it is essentially going to weight higher. It's going to give it a higher weight on a change compared to a third codon position change. And we have these in separate partitions with separate models. And so the ITS changes are going to be different too, right? Because an ITS change from a T to C is not the same as a third codon position and TF1 change from a T to C, which is not the same as a first codon position change from a T to C. So it just makes you put in the full length of the alignment. So you just have to kind of, it'll say 1 to 1. You just sort of manually type in 460 and it tells you what the length is here. And we'll tell it to use a codon specific model. We'll hit back instead of OK. But yeah, that's just the way they name it. And we'll run model test on that as well. And it should run pretty quickly because, again, these are not large data sets. And it has decided that we want the TIM2 with gamma plus I. And for this, said GTR gamma I, which is sort of the most complex evolutionary model, which is usually what ITS gets more often than not because ITS evolves the fastest. It's noisy. It is going to fit the most complex model most of the time. A quick note on models. They're really not very important if you have robust data with robust signal. You can run the exact same tree and you can run it on every possible model and look at the trees one by one and compare them, and the differences are usually going to be pretty minor. And there's actual papers that I can point you to in molecular phylogenetics and evolution of people basically saying most of the time it doesn't really matter. You can usually just go with GTR gamma I. You can usually always just pick the most complex model. It's probably not going to have a strong effect on your data unless you have something really conserved, like small subunit, where there's very few changes, or something, really noisy, where you might need to fit a different type of model, something it's kind of an oddball with weird directional evolution, where you have like, directed evolution changes and transitions and transversions for very specific things. Most of the time, it's not going to matter too much. Go ahead and do it since RaxML does it for you and makes it really easy. There's nothing wrong with ever not doing it, but you don't need to get too hung up on it. We'll go ahead. Do we want to? Let's just run a test tree partly just to see how long this is going to take. So this is still not a very big data set. We have 36 sequences in our ITS data set that's 820 base pairs long. And we have 26 sequences in our TEF1 that's 460 long. But this is significantly bigger than the Pulverilepiota thing. And the thing to know about tree searching algorithms is that the amount of time and, well, the amount of tree space, of hypothetical imaginary tree space that the software has to search to find the best tree is exponentially larger, and it's 2 to the power of the number of sequences that you have in there. So very quickly, numbers get very, very large, and if you have something like 50 sequences in your data set, you have more possible trees than atoms in the universe, which seems insane. So remember that what we're doing here is that we're not exhaustively literally finding the best tree. That's why when we told it to do 20 runs with different starting trees, it found 20 different trees. They were all probably very, very similar to each other, but it sort of ended up in slightly different areas because it is heuristically searching through all of the possible trees to try to find the best one. It's not literally finding the best one. It's trying to find it. And the bigger your data gets, the more it has to search and the more it has to struggle to find that. So let's see what happens here with one tree. One, to glance at it, spot check it, but also just to see and make sure that it runs. And also, I've been getting this problem, which is really annoying. I've reported the issue and never heard anything back from them. I don't know if they really support the software anymore. Sometimes it will tell you that you want to use this model, and then when you try to run the tree, it'll say, that's a bad model. Why did you pick that model? I'm going to quit, and I'm going to terminate your analysis, which is like, well, then why did you tell me to do it? Something I haven't quite figured out yet. Basically, just turn off the gamma parameter and then run it again, and it should work. It's something that is not perfect. It's something we're going to maybe have to try to figure out why it keeps doing this. You can force it to keep running if you ran it in command line, but I'm not the kind of person that likes to run command line because I'm not very good at computers. And if I were better, it would make sense and I could do it. So if you want to, it's kind of awesome. You can use RaxML GUI to basically set up your analysis, and then you can basically export out the commands, export out the partitions, export the models, and then just run it in command line, and it kind of does all the work for you of setting up the run, and then you could run it in command line. Or you could just go, okay, fine, it's good enough. Since these models honestly don't matter very much, I'm just going to go ahead and turn off the rate heterogeneity. especially since we're just doing exploratory tree searching here. So now it says it named it ITS alignment. I should have named it something else because it is indeed ITS and TEF1, but we can see what our tree looks like. Well, nope, that's the best model. That's not what I meant to open. That's not a start tree, best tree. All right, so here's what we got. And so this is an example of one that's not rooted. So it looks kind of funky now, right? We've got some things that are sort of on like the base of the tree. It's just that the software does not know essentially how to display it. If you wanted to display it, oh gosh, you know, wow, that's kind of trippy. Like this is a perfectly reasonable way to display a tree, right? It's kind of funky, you know, you see them. They're not my favorite way to see a tree. But this is, you know, one way to look at it. This is going to be the normal way that we're probably all mostly used to. But so what I'm going to do is I'm going to do Command M to midpoint root it, and then Command U to bottom to top it. And then here we can look at our multi-gene tree now, and I'm going to drag that out of the way. And we can see all of our various clades that should more or less correspond to Husto's Pluteus clades. So we've got the Detrichii clade. Here we have... So actually, I didn't tell it to root it. So here's the thing, actually. I could have told it to root it with the outgroup because this is section Pluteus here. So this actually is functioning as a root. I didn't tell the software to use it as a root. You don't have to tell the software to use it as a root. Sometimes you want to, because when you tell it to make it a root, it makes sure that they are always clustering together. Every once in a while, you'll get something real funky, especially if you're using outgroups that are not closely related to the group you're looking at, because maybe there isn't a closely related group or you don't know what it is. You know, it's a genus that no one really knows where fits in the Agaricales, and you just kind of have to pick something. You might want to tell it. If you did include an outgroup or forgot to specify the outgroup in RaxML, you can just grab the clade here and you can hit reroute. And then it sometimes gets mad at me when I do that, and I don't know why. So maybe I need to select node. Yeah, select node and then hit reroute, and then it'll reroute the tree. So now we have section Pluteus here. And then here's a bunch of sections Cellulodermi. Got the Detrichii clade. We've got I wish I knew my Pluteus taxonomy off the top of my head clade. We've got a bunch of different clades. Here we sort of have the one that we're specifically interested in with this species here that's clustering close to flamans and Arontopes. So we have successfully placed this species within all of the different clades in Pluteus by doing this tree analysis, which is not necessarily something you might be able to do with you know, just pairwise comparisons, right? You see that this Pluteus sequence is, you know, 97% similar to some other Pluteus, but that doesn't necessarily mean it's always going to fit in a clade neatly, right? So this should help you sort that out. Let's go ahead and just for the hell of it, do a run with 20 runs and 100 bootstraps because we can. You see this one, now it's searching each of our individual 20 tree searches, so that it's running them 8 at a time. And they're sort of searching through. It's taking a little bit longer because our data set's bigger. It's still going fast, honestly. So here come our bootstrap trees. And then it's going to give us some support values. While we wait for that, any questions about what we're doing here?
No, this is extremely informative and I appreciate it.
All right. And like I'm doing a Jess sequence validating speed run version, right? Obviously. And
Honestly, all I need. Like,
yeah, because it's not complicated. It's just
no.
knowing the order of operations of what to
Yeah.
export from what and what to click where,
Yeah.
and then also like when it throws that random error, what to do with it, you know.
Like,
and then just for me butting my head up against this for a long time.
like I am decent at computers, so I can troubleshoot stuff as long as I know like what programs to use and how to like order to do it, you know?
Yeah, yeah. Yeah,
Like,
and you'll see various things which we can kind of go into a little bit too. Like I started with the finalized data set. We didn't really start with how to make a data set. And there's definitely a lot. And there's errors that'll pop up in RaxML that'll tell you you have invalid characters in your definition lines. It really doesn't like punctuation. well, good Lord forgive that you have a colon somewhere in there, you know, like it'll get really mad at you, but it'll tell you that it's mad at you for that reason, usually. The annoying thing about that other error we encountered is it just tells you there's an error and it doesn't really tell you what it was. So now let's see what our support tree looks like. We have it here now. It should more or less be the same. We will reroute it. And then put it up here, go to branch labels, label. And in here we have pretty good support values, honestly, for a lot of this. I mean, again, it's ITS and TEF1. It's better than just ITS. And the TEF1, especially as you see with the alignment, there's not a ton of ambiguity in there. It aligns really nicely because it's protein coding. So you get a lot of good signal from protein coding genes. I mean, TEF1, RPB2. RPB1's just harder to work with, but also great. And you see we've got great support values. Which again, these support values are how much signal is in our data. It's not exactly the same as this is the probability that this is the real evolutionary relationship, but this is how strong our data is to say that these two things are related to each other. Here we see our clade here with our unknown Pluteus with 100% support next to these two species. So like we feel pretty good, you know, even though I'm saying that support doesn't mean it's like quote unquote real evolutionary support. It's very strong signal. It's not ambiguous. The placement based on the data we have is strong. And my opinion usually with ITS and especially like ITS plus LSU or TEF1 or RPB2 or something is that, you know, if you have two independent lines of data that are telling you a very strong story, this is pretty good. You know, you don't have to doubt I mean, you know, be shrewd, but you don't have to doubt everything all the time. We can see that, you know, Husto's Pluteus clades also get real strong support. You've got 98 here, one here, one here, one here, one here, one here. You know, we've got strong support for his clades. What we have more middling support on, which is unsurprising given that we only have two regions, is our backbone. This is kind of the called the backbone of the tree. We've got, you know, some strong numbers. Like I think, you know, maybe this Detrichii deceptivus group is kind of a very unique group on its own here. But then we sort of got 80. That's okay. 84 is okay. 66, that's garbage. 57 is just, you know, you may as well collapse that in your mind. Just pretend like that branch doesn't exist. There's basically no support for that. Yeah, it's the way I sort of informally look at it. This is, and honestly, I wouldn't count these 77s for much anything. It's sort of around these 80s that it's sort of like meh, and then 90s and 95 and above, especially. That's like, oh yeah, this is good, or there's a lot of signal in our data to say that these cluster together, if that makes sense. But, you know, mentally collapse these relationships. If this has these two clustered together, but with low value, don't think of them as next to each other in your mind, even though they show up that way on the tree. All right. Well, that's how to make trees. This is how to make RaxML trees, you know, how to make maximum likelihood trees using RaxML GUI. You know, there's lots of different ways to do it. And there's ways to make other trees. But honestly, this is a pretty straightforward way to make very quickly what is essentially a publication rigorous tree. You know, all I would probably do would be I'd run more bootstrap replicates. And one thing I will do with this data is I've got some more data to add to it to try to flesh it out, to add TEF1 for some of these that didn't have it so that there's more equal representation across all of the different regions. So it's not totally finalized, but this is more or less a tree you could plop in a paper. And there are way more, how do I phrase this? There are way lessly rigorous made trees than the one we just did in papers, you know?
Um,
Yep.
were you? I mean, I can go do this on my own later, but it would be interesting to see the cystilepiotas without the like the combined haplotypes,
Yeah,
or
yeah,
pulvera
that would be an interesting way to look at it. Do you have a data set without the haplotypes? I guess we could probably just do it here, right?
Yeah, it's not a clean one,
Let me
so
go ahead and open this up, and we might just be able to kind of delete out the ones that obviously are riddled with polymorphisms. Wow, I've got a lot of things open.
I mean, yeah, that should be fairly obvious. Well, and they'll be, well, you didn't change the names. Well, yeah,
Yes.
any,
Oh
like,
yeah, I've
zero.
got consensus and haplotypes here.
Yeah,
So
there
yeah,
you go.
we can delete these out. We've got, see, that's our out group. We've got this one. Wait, this one, why do I only have haplotype A and B? I'm not a consensus for this one. I might have just done something funny when I was throwing this data set together. And then I also want to go through and I want to remove Steve's ones from Helen. Well, or at least these ones, which are clearly conflated haplotypes, because we've got our R's, our Y's. Looks like one of her collections might be like a purebred, for lack of a better way to put it, I guess.
If it's the PNW05, I would like to see it rerun on Nanopore because I was looking at it and there's some frame shifts in the chromatogram that are just evil. They're
Yeah.
evil.
Okay, here's something where I bet I messed up the naming and named this haplotype B because it has polymorphisms. I wonder if I messed this one up. Because, right, none of your phased haplotypes should have polymorphisms in them, or do some of them. You're muted, Jessica. Sorry.
There's like one that had a couple. It was probably just nanopore noise or something, you know?
Maybe right here in this little string.
But I don't think it was
Right,
that many.
this is different from the other sort of haplotype signal we were seeing. So yeah, maybe we'll go ahead and leave that in because it's
So
not where the other
864
noise was.
one. Let me.
78643.
I can go just check it real quick because I would have noted it. 643.
Yeah, otherwise the only polymorphisms I see are like in Jeff Stallman's Hawaiian one, and our out group has one, which is fine. That's not gonna influence this analysis.
Yeah, the haplotype B did have a couple ambiguities in it. For that one you were looking at.
Okay.
When I split out the haplotypes, it wasn't a very high. It was an 80 reads and consensus file, but like one of the haplotypes was like it was like one of those like 60/40 ones. So when I split out one of them, it only had like 20 or 30 reads, and so
Okay. So it's
yeah,
sort of like there's
there might
definitely,
be some noise in there.
yeah. But I think that where the noise isn't actually gonna probably affect this too very much. So let's see where to go. Here's our no haplotypes. Let's see if we can see if it tells us anything different. See if we get some more information here. Ta-da, we've got a model. Let's go ahead and run it with bootstraps, because why not? It's so fast. It's such a small data set. I'm not too worried about it. Boom, boom, boom. All right. That's the wrong one.
Like the thing that was killing me with nanopores, I couldn't even figure out how to get it to do bootstraps with maximum likelihood. Not on nanopore, Eugene.
With Eugene.
Yeah,
Yeah.
it maybe the settings in there, but I could not find it.
I think it has settings, but I also don't know how they work, and there's not a lot of transparency in it.
Yeah.
Okay, honestly, this is not telling us almost anything different than what we had seen. Kind of telling us the same situation here. So this is kind of an interesting topology, which probably tells us more about we don't have a ton of signal in our data than it is telling us about a real topology. So like, see how this ECV2572 is literally like on the root almost of everything else?
Mm-hmm.
What we really probably have here is all of these ones are sort of kind of clustering together, but the algorithm doesn't know how to relate them to these other twos because they're not necessarily closer to the European sequences or closer to this other cluster. So it's kind of given us an ugly topography here. But this is just a topography that basically tells you kind of what we knew. Things are messy here, and there's not a really obvious strong signal. There is clearly a cluster, and a cluster, but we don't know what to make of it. What's interesting is that when we have our haplotypes phased, is that we have this and this, so we have one mushroom with both of these clusters of haplotypes in them, right? Which is what we knew. I mean, this is just showing us what we did kind of know already through what you had done through going through sorting all of these, but like we are clearly seeing this sort of like whatever's going on here, there are two distinct sequence variants or haplotypes that are interestingly at least consistent across 3 collections. Which is really telling us that something odd is happening here, that it's actually more common in our limited sampling, which is pretty limited. It's more common to see something with both sequence variants than it is to see something that only has one, right?
Yeah, well, I want to go back and make like a better tree for those entelomas I was looking at because we have more of those that are just one or the other. Like there we
Yeah.
act because like on this one we actually don't have a clean read of PNW07. Like
Is
there's
that which
no
one is PNW07? Is that this one?
uh
I should have named them differently.
yeah
That's right, actually, because I think this Renee's is I think that one also actually
that might be
might
the PNW05.
I think it
That's the
has
PNW05 because you said it's Renee's, it's the PNW05.
yeah, I think that one actually maybe we would have wanted to exclude because I think it's conflated a bit,
Yeah.
but
So
yeah.
yeah, it is. That's the one that needs a
Oh, is that the one
rerun.
you were looking at with all the frame shift
Yeah.
Tendels and stuff? Oh,
Yeah. So I don't
salvage it with one or with like one frame shift in each direction
the
manually if you really
forward
want to,
had
but
one frame shift. The reverse had at least two. So when I realized there was a second frame shift, I'm like, I can't fix this.
yeah,
So
yeah,
because when there's
it gets
one yeah,
yeah.
anyway, that's chromatogram. Yeah.
I manually unshifted a bunch of chromatograms in my day, and where your brain is thinking about the shadow hump, right, and ignoring
Yeah.
it.
Yeah.
But
And
then
I
at a
got it.
certain point, you might hit that second frame shift, and then it's like, I can't think in both 1 and 2 humps. It just,
Yeah.
ah.
Yeah, well, because I got like forward, like basically done until it turned into noise. I was like, oh, I'm so proud of myself. And then I started doing the reverse, and I got like, I don't know, 30 base pairs in, and it's like another frame shift.
Yeah. And then, like, usually all the best you can do there is fill all of that gap in with ends in the middle, which,
I mean, I could have, but
well, in this case, you're actually trying to get each individual haplotype, so that wouldn't even help you. That would give you a
yeah,
consensus sequence, right? But yeah,
yeah, that's not. I was trying
yeah,
to at least like get something approaching haplotypes, but
yeah. You
um,
kind of just can't with a Sanger read like that. It's kind of impossible.
but yeah, I want to look at those entelomas because like for the entelomas, we do have some of one that are only one sequence, and then we have some of the other that are only one, haplotype.
Yeah,
Versus this one where there's literally a single CaO3 that I found that we've found that only has one haplotype. The rest are either Sanger reads, GenBank reads, or they have both. So
yeah. Yeah, so yeah, I don't know what that actually means in biological reality here. I mean, we can see it in the tree, we can see it sort of with the haplotypes. Does that mean this is all one species, including the European species? Who
I
knows?
mean, I don't know
I
if you
mean,
were
this data isn't going to answer those questions for us right
yeah.
now yet. This would be a great candidate for some other gene regions. Be very interesting to
Yeah.
see if TEF1 and RPB2 have two very distinctive haplotypes. This is an ITS specific thing where sometimes you get problems, or not problems, but you get weird genomic issues with the proof copy, the proofreading, ribosomal array where you could get two distinct haplotypes entrenched in a population. That can happen. Like there's weird genetic reasons that this could actually just be one species with two discrete variants that are evolving consistently and independently. I mean, I don't know.
Fungi are weird.
Fungi are weird. And this is what
I
part of what makes it interesting is weird little things like this.
yeah.
This is a fun situation, though, where no matter what happens, whether we consider the European species the same or not, it all goes by the North American name. Thank you, Merle. We got Ptaciformis, published in 1912 from woods near Seattle, which is surely a parking lot now. Because I'm sure
Yeah.
Seattle was a lot smaller in 1912. But yeah, that was from his train trip to do his Western collections. He published a lot of lepiotoids from Seattle. A lot of cool species from up there, and then went down to California and also published a bunch from down there.
Nice.
And he
Yeah,
caught
this
a
is
lot of good
super
things.
informative, Josh. Thank you so much.
Do we want to go sort of work backward to look at how I organize the sequence data to make these FASTA files?
If you want. So you're doing it in Fig Tree? No.
So let's go. Oh gosh, what's a good one? Let's look at the Phaeolus one because that actually is relatively manageable. So here is how I organize my data sets, basically in spreadsheets, in Google Sheets, where I have exported all of the data from GenBank and then put it into a spreadsheet, and then semi-manually added the micromap data, because obviously none of these databases talk to each other or format all their stuff the same, so you kind of have to fit it together a little bit. And then I also have it sorted so that you can basically go here and then you can just look at the LSU sequences if you wanted to pull out just the LSUs or RPB2 or something like that. Honestly, in this one, I think I only did ITS because it was the only thing that I only fully processed the ITS because it was the only thing that was interesting. But essentially, I have it, concatenate my FASTA names from these fields, and that's a way that I can really help try to make sure that these are all consistent. But if you notice already, somebody put UBC F20358, and then someone else put UBC space F35895, and that's what can get annoying. These aren't the exact same specimen, but sometimes you have the same specimen and you have these space problems, and you have to kind of go through and manually fix them. The way I get this data is a bit uglier. This is where maybe people like David will tell me, I cannot believe that you were brute forcing your way through like some of the ugliest like data management back of the envelope, not back of the envelope, but just like the ugliest way of doing something possible. As if we go here to Phaeolus, I'm just going to kind of recreate this one real quick. And then usually put in brackets organism, because sometimes it'll be Phaeolus, but it'll be picking up the fact that like Phaeolus is a name in the paper that the sequence was published in. Like it's not, instead of searching every field for Phaeolus, you want it to just be like, no, I only want the things that are organisms. So see, it dropped by 8 random things. Do a quick look to see if we have anything crazy here. Normally, I like to have it I'm gonna have it do that. Sort by sequence length so that I can see. I mean, that's big, but that's like a full RPB2. Make sure there's no like genomes in here, because if you try to export a genome, it's gonna get real weird, and it's not gonna fit the data structure of everything else. Sometimes you'll get a whole like meta barcoding ITS from a soil sample, and you'll have to like basically do things like not Illumina. And then it'll filter out anything that's Illumina, right? So if you have a bunch of that noise, you can filter it out in order like not genome or not scaffold or not OTU. You might want to do OTU a lot, because sometimes things, even though it's only supposed to bring up things that are specimen based, it may bring up a lot of craziness here. So what I do, and again, don't take this as like this is a good way to do it, it's just the way I've gotten it to work, is download this INSD seek XML. It will create a file. We open that up. We go to XML to CSV converter, and we choose a file. And we put that file in there. And we save it. and now we have a CSV. So now we have turned that into something that at least Google Drive is going to be able to read. We'll pop it in there. It's funny. That's not the same name, but it test file. Plop this in, open with Sheets. And here's where we have to sort through the giant mess that is trying to take an XML format and put it into a flat spreadsheet where we've got blah, 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 all kinds of craziness, right? So, you know, it has all of the data we need. We just need to figure out how to get the data out of there. And this is where, thank you, Sigrid, for pulling me into that Sporormeal paper, because I basically figured out how to do this for that, because I needed to get a whole bunch of sequences organized out of GenBank into something variable, or into something usable. So basically, here is what I want to be my fields. This is what I basically need my fields to be. And I'm going to go ahead and just literally copy these over. We've got our definition line. We'll want to copy that over. This will help us in, not bold, paste. There we go. We want our sequence source. This would be the genus and species name, more or less, sort of, kind of. And then we want our sequence. Okay. So that's the easy part. Now here, oh, God, tell it to do that so you can see what you're doing, or at least that's what I do. So now we're going to use XLOOKUP to essentially pull the fields that we are interested in. I need to close some of these, or else I'll get confused. So here, our tab name is test file. Well, for best results, no. Okay, here, just let that throw an error real quick. We're going to rename that without a space in it, because that's just going to make my life easier, even though you can get around it. We're going to tell it to pull test and test. And then we're going to look at our final two fields. We got IO and IP. And the first one is going to be our second to last column. The second one is going to be our last column. So what it's going to do now is I'm going to tell it, look up geolocation name. And so it's going to find the column with geolocate, or geo underscore LOC underscore name. Why can't I paste? Wow, don't do that. Here, just sorry, shouldn't be talking and trying to do this at the same time. All right, there we go. There we go. So now it's going to go find basically the line that says the next value is going to be the location, and then it's going to pull that location over. Then you do the same thing with strain, which not everything will have one, but a lot of things will. Oh, I'm silly in the way I pasted that. Don't worry. We're just going to go ahead and delete all these out just so it's easier to navigate. Okay, so now we can see our different strains. Now we're going to see our specimen vouchers here. Oh, why did I do that? Isolate. And here's really, you'll start to see why I get really annoyed at the way people do stuff. Isolation source, because what will happen is you may have the voucher number of literally an INAT observation or an herbarium specimen or anything, and it may be under strain, maybe under specimen voucher, it may be under isolate, it may be under isolation source, and it may be under biomaterial. And that's really annoying. And sometimes for the exact same specimen deposited by the same person, but in two different batches, they may put the collection number under specimen voucher, they may put it under isolation source, they may put it under biomaterial. So in order to be able to make a concatenated data set, we need them to have the same name, and we need to know that they're actually from the same source. So we actually kind of have to be able to check through all of these, which is just so annoying. It's a lot bigger than it needs to be. So for example, here's AFTAL ID 702. Here's AFTAL ID 702. Here's AFTAL ID 702. So these are all the same thing, right? This one has a specimen voucher called AWWSN in parentheses CUW New York. This one has ANWSN under strain. This one has a different version of this because it doesn't say New York, right? Those are all from the same specimen. Every single one of them is slightly different. So what we're going to do,
And the New York
gonna-
one doesn't have location over in the location.
and it doesn't have location. This one doesn't have location. Oh, this one says Andy Wilson. I didn't know that was Andy Wilson's. This one's Andy Wilson S in New York over here. Honestly, none of them have that the location is New York. It's just kind of like it's all over the place. And that was Brandon Matheny who uploaded those. So, you know. Basically, what I'm going to do is now that I've gone through, I've pasted all these as values, right? So it's no longer that L, it's no longer the X lookup. So now I can essentially be like, you know what? I want to call all of these this. And I don't want to have to deal with any of the rest of this nonsense. I'm just going to go ahead and delete this. And in fact, what I normally do is I kind of see here, you know, right here, these people put die under specimen voucher. For some reason, they decided to put under strain here and then put the herbarium code under specimen voucher. But this die is the same one as the one above it. You can't necessarily tell immediately because of the way they've decided to enter it in here. Oh, and also, actually, that's also part of it too, is these ones that are ref sequences of the type, it creates a duplicate record, but it formats it differently than the source. And it's just kind of annoying. So basically, I kind of manually go through here and clean this up. The bigger your data set is, the more annoying it is to go through and to clean this up. And sometimes you just look at stuff and be like, I don't really care about this. Or like, look, this is PS21 Tom, small subunit. Actually, no, that's a full read. But I go down here and I'm like, 248. That looks like an Illumina code, probably from some MetaBeat barcoding thing. This is an Illumina code. This might be a real voucher, might not, but these are really short sequences. I don't even want to bother cleaning them up. I'm just going to delete them out of my data set. But I definitely want to keep this Jonathan Frank one. You know, there's a lot of, this is a part that I'm still just kind of doing manually until I can find a better way to do it. Which I'm sure there are much better ways for people that are good at computers. And like I said, this is a relatively small one, but you kind of got to go through here and then just sort of fix them all up. And then you got to go and be like, oh my gosh, this HHB dash, this HBB doesn't have a dash. I've got to delete the dashes out. But to give you a rough idea, that's what I do to get data out of GenBank into a data set.
I mean, you could
Micromap
you could
is
probably code like a bunch of Excel prompts to actually like, search for the voucher number in each like does this search for this voucher number in each column and then if it does but it
But then the fact that the formatting's different, some have spaces, some have dashes, some have
yeah.
underlines, some have colons, and it's like that. And sometimes yes, that's what makes it so like there's no like perfect, I don't know that there's a perfect
No.
way to automate that, but there's probably a way to clean it up a lot so that you're doing a lot less manual work. You're still having to do some. Okay, so here's how I get data out of Micromap. And I don't know if I'm doing this right, but I think this is what Steve told me to do, and it's frankly a bit confusing. Go to any random project, doesn't matter what, type in what you want, Phaeolus. You have to put a country, so it's annoying. You may have to go in and put United States or put in Canada. Then say not in project, verified, refresh, and we'll see if it'll. And this usually works. Sometimes even then I realize I've forgotten to do something right. And we let it think for a second. And now it's not searching in this project for Phaeolus from the United States. It's searching in everything that's not in this project that's a Phaeolus from the United States. So you can just make a dummy project with nothing in it or one thing in it, and you can use that to search. Feel like this doesn't make a lot of sense, but this is the way it is. Then what I do like is that his exports are relatively, well, no, I was going to say a relatively clean CSV, but it exports a CSV. And we can open that up. Forgive me for not having Excel, so we're just going to kind of look at it in numbers, which is, you know, a thing that one can use, but it's kind of confusing. But this is at least a relatively sane data set compared to the XML format that we get out of, right? You can look at this and at least make sense of it pretty immediately. But there's weird stuff in here that I don't understand. For example, here is the same INAT observation with, you know, all of these different things and identical sequences for all of them. I don't know why that is. So you just go through and delete the duplicates out. I don't know why, but it does that where you get the same thing multiple times. My only guess is maybe that it's to do with like how many sequences were originally clustered in MycoMap, but then it overwrites them with the real one.
Likely
I'm
what's
Go ahead, Scott.
Yeah, I have a feeling. So something weird that MycoMap does is, okay, let's say you get your Kyle uploads a run, he's got a sequence attached to it, and then you do the whole import to INAT thing. Once you import it to INAT at some later point of like a refreshing of external data, a lot of the times that sequence box will have a duplicate sequence of it, reflecting that it's also in INAT, a part of the INAT observation. So not only do you have the sequence that was imported into MycoMap for that observation, but then it also pulls that identical sequence that imported back in. And so now your S equals 2 in that box.
Well, also,
Or 6 or
some of
what.
these things may have been sequenced multiple times. So because some people send out splits to multiple people, right? So if multiple sequences, like unique sequences were generated, it's going to attach all of those to the observation, even if they're the exact same code. Like,
Yeah, that could definitely happen. I
yeah.
don't think that explains why there's 5 of this same one. I just don't know what's up with that. I just go
Well,
through and change
I've
it.
seen for some of Steven's reruns, he did like a rerun of full length ITS, but then he did a rerun of
Oh.
the same stuff
Oh.
for full length ITS plus just ITS2. So that would produce at least 3
But these are
sequences.
all just ITS. This is the exact same, but like it's literally, it says it's the same. And then you get this weird thing where you get a sequence 2, and I don't know, like, I don't know what all of this means. Like I said, I'm kind of brute forcing data out of MycoMap, and I don't know that using it for something that I don't know that Steve clearly had this intention in mind for the export function, or I don't know what he does with it, or if it was sort of an idea that then got sort of abandoned at some point, and now it's sort of half functional, you know, but it
Th-
does get data out.
There's a lot of ability to duplicate sequences through unreconciled workflows on the site. You can, through some workflows, make 20 or 30 copies of the sequence and get it loaded in. And then stuff like the second read are cases where they recognize it and try to add multiple data instead of having a reconciliation workflow. I put feature requests in for a lot of these things.
Okay. That would be very helpful for at least what my purposes are, you know, for using it. Yeah, it's clearly that there's just some stuff here. And, you know, MycoMap, we love it, but it also drives us crazy sometimes. It's the best tool we have, but that doesn't always mean it's, you know, it's not,
It's a
it's just,
small
you know.
indie website.
It's a small indie website and it's amazing for what it, I mean, I
Yeah.
can't imagine trying to make this, right? So, you know, it is amazing. It is just that, yeah, we kind of end up with stuff like this where you go, okay, I'm literally just going to like sort these, or even if it was long enough, like just tell it to only report, you know, unique values. And then I could just kind of thin it out that way. So it works. I would just delete out those and then I would paste them into this spreadsheet to basically just fit them in if I had 50 of them or something. And I would just you could put a MycoMap number here or something. You don't have to fill in all of these fields, but you want the sequence, you want a specimen voucher, like an INAT number. And then I also like to have location, and I'll reformat these usually to simplify them, you know, from sometimes you don't need all of this. If all of this information is in your FASTA, that means it's also then in your tree, and you've got these really long, you can just shorten that down to Russia if you don't care where in Russia it was. You might, because you might want to know whether it's far east Russia near Japan, or you might want to know whether it's St. Petersburg, that might be important. It might not be. In this case, I'm just gonna, I might just call it Russia. I usually do, the states, because I care about the states, because that's where I am. So that's how I make data sets, which takes some work. I mean, there's other things to do here too, right, that I didn't go over. So one of the most important things you're going to need to do here, right, is you're going to need to sort these into locus, because that's obviously very important, because you can't align ITS and TEF1 and LSU all together. So I'm going to make, a locus field here, and I'm going to filter by polymerase to be like, okay, yep, here's my RPB2s, and then I'm gonna filter by. The only thing that gets a little tough is that ITS, LSU, and small subunit, right? Those are all in different combinations. Sometimes, like, most of the definition lines of ITS contain the words large subunit, or 25S, or 28S. Again, there's probably a way to try to make this happen a little faster, because if I just do large subunit, I'm going to get both just large subunits, and I'm going to get the ITSs. But this does help filter it out quite a bit, right? See, I can immediately go like, well, these are all large subunit. And when they're sorted by length, that really helps also, because usually the large subunits are longer than the ITSs, but not always. For example, here's some really short large subunits. And then also another thing that I do when I have one of these that's really long, so it's like 1500, I do ITS plus, because it is both probably ITS and LSU. And so I will actually include that in both my ITS alignment and my LSU alignment. And I'll trim, you trim off those long edges. You'll trim off the ITS part of your LSU alignment, and you'll trim off the LSU part of your ITS alignment. But that way you get to use all the data you have. I guess alternatively, you could copy it and I guess just like insert the same, and then call one of them ITS, call one of them LCM. That would also be fine. You can do that if you want. Whatever you want to do. And then, yeah, like I said, clean all this up, which we don't really need to go through right now. But actually, you can clear some of this out. None of these have isolation source as the collection number. And almost none of them have isolation source as the collection number. But every once in a while, they do. And then the other thing to do is to go down here, and here's, you could recreate this obviously with just a little playing around with the concatenate function, but you essentially just tell it, hey, I need you to grab, let's see, I need you to grab my specimen voucher field here, which actually is I2. So that's perfect. Oh, wait, no. No, I2, we'll call it I7 because it's sorted. And then tell it to grab after that the species name, should be E2. And then if it has a location, it's B7. If it has a location, well, if it doesn't have a location, don't put anything. If it does have one, put underscore location. This would be if there was a type field. I'll often usually, oh, I didn't do it. I didn't go through the actual pulling it, but K2 would be here was where you'd put if there was a type or not. And then you put your sequence string here. I keep doing 2 and 7. I'm confusing myself here because I'm kind of trying to half do this instead of fully do it in the sake of time. So my apologies. What did I do wrong here? No. No. I feel like I accidentally deleted a X lookup. I don't know what I'm doing wrong here. I'm doing something wrong. Well, it would work. We could debug it if we wanted to, but does that make sense? At least what I am doing.
Yeah.
Yeah. All right. So that maybe we should have started with this and then gone to the phylogenetics, but honestly, this is like a worse, crappier process. And I wanted to start with the fun making trees because it is a lot more fun than dealing with all these weird sort of data formats and compiling everything into a sane structure and then trying to get it all
Yeah.
organized correctly.
Well, and honestly, like for some of like the really short, like I'm just looking at some stuff on INAT and I want to like, see a visual representation to make sure I'm thinking the right thing. Like it's like maybe 10 sequences max and I can honestly just like manually paste those into Eugene and call it a day.
Yeah, you
It's
could just manually make it a little faster real quick or
yeah.
find a way to export them out. That's really targeted toward
Mhm.
exactly what you want.
Cuz you can go right to the blast page and download a FASTA right off the blast page.
Yes, yeah, that's a really, yeah, if you're just looking at,
So,
hey, what are these 10,
yeah.
what are these most common things right here?
Yeah.
Yeah,
So,
that would be fun. Can we just do that off of like the Nama 20?
just pick one of the observations and here, let's find those entalomas I was losing my mind over. Uh, where's the putative hybrid project? Hmm. Now, the one thing I will say is this is not going to pull any of the um, phased out haplotypes with the FASTA, just because Micromap isn't really set up to pull those. Like this is a really recent thing we started doing,
Yeah. Yeah. So
but
really
um,
we're we're like I said, we're kind of brute forcing in the things we want to do in ways that I know could be more eloquent with a lot of work put into sort of
yeah.
optimizing them. But
But anyway, I just linked the blast page for one of those entalomas.
okay, let's open that up. And let's see how this works. Because I think I have done this a long time ago, sort of downloading straight out of that. You can also download straight out of NCBI Blast as well. So if you can do the same thing. You'll
Yeah.
probably have less mushroom relevant data. Um,
But for like a quick and dirty tree, like not a paper
but for a
tree,
real
like
quick
this is
dirty tree, just to see
yeah.
if like is 36 and CA4 really different, we can probably just open it up and do that real
Yeah,
quick.
there is going to be a lot of duplicate data for some of these between like GenBank and the local blast because of
Yeah, that's
us
another thing
uploading stuff.
that's kind of another crappy thing. That is something that I do like about the CSV exports that you do get out of here is that it does export out when present, and it's not always been back referenced.
Ooh.
But when it does have the GenBank exception, so it can help you not duplicate your data.
Yeah, I don't think
There
does
are
that.
nice things that it does, even if they're not all perfect. Let's see, where did that? I already have Eugene up, so that's why. God, I gotta close some stuff. I get stressed out when I have lots of things open.
I feel that.
Do a quick math. Math. Pretty sane so far. Yep, everything looks in the correct alignment. That is something that I do get out of the micromap export that I don't know if that's a bug or not, or something that ought to be fixed. It isn't. Sometimes I get things that aren't reverse complemented, and I don't know if that's because the original wasn't reverse complemented and it didn't overwrite what I get on the export page out with the reverse complement or what, but I do have to not often, but sometimes manually reverse complement things from the micromap export.
Micromap has gone through some periods where it wasn't automatically reverse complementing things.
Maybe that's what it is.
Yeah.
Yeah. If you did want to do just here, by the way, in PhyML and maximum likelihood, here is where you would tell it how many bootstraps to do. Because yeah, you'd probably want to tell it to do bootstraps, but it's probably going to do Felsenstein bootstraps, which are different than the ones we're doing, and they're
Yeah.
probably going to take a lot longer. And that's one of the main things that I don't like using this is that it doesn't automatically search for models for you. You have to manually put them in here,
Yeah.
and it doesn't run fast. at least in my experience, it's very slow compared to RaxML, which will run it in parallel. So I've got 8 different workers, searching through tree space simultaneously. So it's a lot nicer than having to deal with that, especially when it's as simple as a couple right clicks.
Mm-hmm.
And I'm just gonna throw it on the desktop.
Is that just the GenBank? Or is that both?
That one?
Is that both of them? Or just GenBank? There.
Sorry, that does what?
Never mind. I just don't know. I don't know what I
Sorry,
was asking.
I literally was like, here I am clicking buttons, and then all of a sudden I forgot literally what I was saying the second before. So let's see what we get in Raxamel for our little Entaloma family. So here's an example of an issue. Illegal characters, it's really mad about these colons. I think that's mainly the problem, or there's spaces, or blah, 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 blah. There's quotes. It's really unhappy with this. What's really nice about this Raxamel GUI is that it very politely will make a Entaloma modified .fasta, and it will kind of like fix that problem for you. There's certain special characters it won't like, it'll just get really mad about. So I normally go through and I remove all the characters using like Control F, replace in my Google spreadsheet. I just replace all the colons with underscores, replace all the spaces with underscores, remove all the quote symbols, remove all the periods, that kind of stuff. But um, it will at least try to do this. Let's see. Let's do a sanity check tree before we do too much. Oh, this is going to make a mess of my desktop. Never mind. We're going to make a new folder because I
Well,
don't want to.
you might need to run the model test again.
Oh, you're right. I need to run the model test, period. So, there we go. How many sequences do we have in here? We have 97. As you can tell, it's slower. It's noticeably slower, so it's going to take a minute. It starts with the more complex models, and then it runs the simpler models, and those get quicker as it goes on. But yeah, this one's going to take not a long time. It's going to take just a little bit of time. Let's see, untitled folder. That's where I want you to be. So, no, not too bad. Took a moment. You'll notice that sometimes the name of the model might be different than what it auto-populates up here. I don't know. It's either the same model or it's one that's essentially the same. And now we should be saving it to untitled folder, I believe. Which is good. So let's just go ahead and do a single tree. That was fast. Okay, I thought it was going to be longer, honestly. Let's go ahead and take a look at our... Don't ask me why. Sometimes it calls it last tree. Sometimes it calls it best tree. I don't know. I don't know what that's about. So here's all of the things that came up the closest on Mycoblast. As you can tell, we've got a whole bunch of stuff up here, right? None of this we actually care about, right? We've got all kinds of other species that just still came up in the blast as somewhat similar. We've got Velutinum, which is one of the most beautiful mushrooms. We've got some other Entalomas that are also probably beautiful. I'm going to collapse them just to get them out of the way so that we can focus on the meat of the question here. So we have things we don't care about. We got versicolor, fusco squamosum, corycis cerulopodium. These are the ones we're interested in, right?
Yeah, it should be the Folia contusum Vardia discolor um and then the CA36 I think used to be it's Folio contusum var discolor basically. And then I want to whatever the other one is probably the CA04.
Yeah, so here's the CA36.
Or maybe
Here's
CA48. I'm not sure. I
here's the
might need
disc.
to actually make just a proper data set for this to look, because
Yeah, we could probably clean this up a little bit, but here we have pulled out all of those here. We've got a 33 here that's kind of sitting in a funny spot. I wonder what
Yeah.
that means. We've got CaO4, which is a big cluster here.
I think 48 was the other one, and I don't even see 48, so I'm not sure
That's not.
why it didn't even pull.
Those have 48s in them, but they're not Ca48. I don't know if it pulled Ca48.
That's weird.
And I hope there's not a problem with the blast export because this is a handy little cut. It also could have potentially trimmed the 48 out of some of these because some of these look a little
Yeah.
odd. They've got a GenBank number, then like a double underscore, and then just the fundus number. So I think something about the export, or actually, let's check it. The export might be fine. It may be what Fig or what RaxML when how it modified it, it might have modified it and cut off what we actually wanted. That's also possible. Wait, where'd that go?
Thought it was 48. Let me double check. Yeah, CA48 should be one of them. But but the thing is, like I said, it's probably mostly pulling the provisional species names, and that is probably what you're seeing because we moved to doing the species, like the GenBank format for the names for new
Oh.
temp codes. So it's a single quote, sp dot single quote temp. Because I'm not seeing so all of these that are kind of weird are probably the hybrids.
Yeah. Yeah, so it might take a little manual renaming of your data set
Yeah,
there. And yeah,
good to know.
that's something we can figure out, right? And like I said, this part, especially the getting data out of GenBank and getting data out of Micromap are things that I bet there's better ways to do this, and someone
Yeah.
can tell me what they are. I hope at least, because I don't mind it. I can, I can pull, especially for something small like Phaeolus. I did this in an hour maybe, but from downloading it, getting the Micromap stuff, putting it all in the spreadsheet, sorting it, making the alignment, making that tree that I posted to
But
Facebook. I
doing
can do that in like an hour, but it's still an hour.
Mycena?
Oh yeah, no, I do this with I did this with all Pluteus, and that took me hours.
Nope.
It was all Paniolus, and that took me hours too.
Cool.
I
Well,
mean, it is actually really fun. So the one thing that is really fun about doing all of them, though, and I'm just going to show you this because this is where I start to go like, this is why it was worth it, is to go here to actually let's use the Pluteus or the Paniolus data set as an example. It's because when you do actually get the data set, it's kind of like, you gotta be kidding me. Like, here is every Pluteus on
oh,
GenBank and Micromap.
that's crazy.
And or Paniolus, sorry, Paniolus. And it's really exciting because you start going like, wow, I can totally see my species. That whole clade is Phenicecii. This whole clade is Cinctulus. And like we were talking about, Scott, maybe that's something that like Cinctulus has these two sequence variants that are like consistently, maybe what we're seeing with the Pulvera lepiota, where there's like two consistent sequence variants that are shared, right? They don't separate out into two species. They just have them.
Yeah. I kind of want to go do something on Neolentinus.
Ooh,
Cause,
Neolentinus would be fun. Oh,
cause,
especially with so many crazy haplotypes.
yeah, those are all polymorphic. All of
All
them.
right, Josh, I have a question. So hypothetically, let's say somebody starts running nanopore on another gene region, like a protein coding region, and you see two distinct haplotypes. Would that be like sufficient evidence for hybridization, do you think?
It's ever I it's not necessarily. It depends on how different they are, right?
And in what part of the like intron versus exon?
Yeah, if they're in the introns, I'd just ignore them, more or less, right? Because that's the protein coding introns are typically even junkier than ITSs because ITS isn't, it does have a folding structure, it does have constraints on its evolution. There's a lot, not all of the intron, but parts of the intron that are just literally just random evolution, you know. That would be helpful, like within a single species, you could look at the introns and get some potentially some interesting information there. In the exons, yeah, that's where you have evolutionary pressure, obviously, sort of acting on them. And if you saw a lot of haplotype variation in the exons, if you had a lot of haplotype variation in the ITS, and you could phase them out, you could definitely come up with two putative parental types. If you could match them, you know, if you have other data to compare them to match them to be like, not only does the ITS match this other species, the TEV1 also matches like between these two species, and RPB2 does, then yeah, then you're really mounting a case of like, either two species and this is a hybrid, or one species with some weird stuff going on, you know, still in the realm of there's some weird stuff going on that's not normal. It would
Yeah.
probably, it would definitely be, right, you know, it could definitely be evidence for hybridization, but it's not the only thing. It's perhaps, but perhaps the more likely thing if you've got it kind of nicely separated consistently into these two parental types.